Is shedding of the vaccine material possible? Like, it sounds good. And this is the information I've been studying for the last couple months, because this is one of the most common questions that is being asked by the online audience. And this has started because of the fact that women notice that their exposure to people who were vaccinated affected their menstrual cycle. And this has led to the possibility amongst the public that vaccine material could be shed. And of course, no one really at the moment has a scientific proof of this. And I wanted to see what kind of scientific information is available to study this, whether at least it could be theoretically possible. And over the studies, I'm going to spread this material into 3 videos. The first video, which is the one I'm going to be talking about today, is going to discuss, is it possible to shed vaccine material or not, theoretically, and specifically material that obviously can lead to some sort of immune response or biological response in an exposed individual. And because of this, meaning through mere exposure to someone who's vaccinated, if there is a biological response in the untreated individual because of such exposure, there are limited options as to what that biological material could be. And the one that I have been focused on, and this is what we're going to be talking about, is exosomes via breath. So I've discussed exosomes in the past. Let's quickly review that information. Basically, exosomes are tiny fragments of cells that can be shed by a cell, and they contain biological materials such as RNA and proteins, and they can circulate. And this is one of the ways how different cells communicate with each other in the body. It's actually, exosomes can act, think of it as one of molecular languages, one of many molecular languages that our body uses to talk with each other. So that's exosomes. In a second video, we're going to be talking about what has been discovered in relation to exosomes and in vaccinated individuals. Specifically, we're going to be talking about mRNA vaccinated individuals. Let's show you these cool paintings. And in a third video in a series, we're going to talk about exosomes that we have learned from COVID-19 patients, which provides the greatest body of evidence of our understanding about exosomes with relation to containing spike protein and the SARS-CoV-2 viral material. Okay, so my name is Dr. Mikola Rashek of Genomics, and let's get started in the first installment. So right away, let's perhaps define what shedding is. And for that definition, specifically, I looked at the FDA document that is a guidance document how shedding should be studied in clinical analysis. And this document is from 2015. That's the latest one I was able to find. Don't know if there is an updated version. Perhaps there is, but I couldn't find it. So let's talk about the one I did find and studied. And the way FDA defines shedding is transfer of virus or bacterial gene therapy products or oncolytic products, we're going to ignore that for now, from treated individual to untreated individual via either fluids such as saliva, nasopharyngeal fluids, so those that come from your nasal cavity, also or through skin wounds or pustules. So basically, or feces as well. So these are the transfer of products. However, notice breath, which is of interest to us, is not mentioned there at all. And in the entire document, neither were exosomes or extracellular vesicles, which defines the family of different cellular fragments that cells can release. Exosomes belong to this family. Basically, exosomes are the smallest of the bunch. And you can think of exosomes almost like virus size. They actually are coronavirus size, and they are not mentioned at all. But let's talk about for a moment, and this actually might reflect the fact that this document is not properly updated and does not reflect the latest emerging science with regards to exosomes being shed via breath, which is what we're going to be talking about in today's video. So I would not be surprised as if eventually this document was actually indeed updated and included exosomes one day in the future. Does not include right now, but that's basically what we have to deal with. But let's talk about what does virus or bacteria-derived gene products mean in this case. So what are the gene products according to the definition of that document itself? This is very interesting because basically that means any genetic material that is being injected or basically transferred into an individual that will result in one of the following. It will either be transcribed, it means that the genetic material is then used to produce RNA, or it will be translated, which means RNA is being used as a template to produce a protein. That's what mRNAs do, and this is what mRNA vaccines achieve. They insert template into the cells that can then be used by those cells to produce protein of interest, in this case, spike protein. This is why these vaccine injections are referred to as vaccines, because they produce spike protein that leads to immune response. Or the genetic material is inserted into the genome of the cells, and this could happen either in vivo, meaning it happens directly inside my body, or ex vivo, meaning my cells are taken out of my body, they're being genetically manipulated, and they're being then reinserted back into my body. And this is what is being done with some of the cancer therapies. So by this definition of gene therapy, the mRNA vaccines completely fit that definition, because we're talking about genetic material that is being translated. So we're talking about introduction of genetic material in the form of mRNA, which is being then translated into a protein, the spike protein, and it is being introduced into a human for that purpose, and has a direct in vivo impact inside our cells. So I've been quite hesitant in the past to acknowledge that that mRNA vaccines should be called gene therapy products, but clearly by the FDA's own definition on what gene therapy products are, it clearly fits that definition. So many individuals have been using that definition. I was reluctant, but it completely fits. Okay, why was I reluctant? Because traditionally I think of gene therapy as genetic material that is being inserted into the genome, and then it has an effect. However, mRNA vaccines are also the first of its kind. We've never used this type of technology in the past in order to attempt to either treat or protect ourselves from potential infection by any pathological agent. So perhaps indeed we are now talking about a new form of gene therapy in the form of mRNA vaccines. So I thought that was interesting. But furthermore, when this document discussed is when should shedding analysis in preclinical studies happen. And the document mentioned that if humans are being exposed to a product they've never been exposed to before for therapeutic purposes, well, then they should be analyzed for potential shedding, or option number two, also humans were never exposed to such product, and on top of that, the administration route, so how you insert the product into the body, does not reflect on what happens naturally, which is exactly what happens with these vaccines. We are injecting these vaccines, and yet the actual infection takes place in our nasal cavity. So another reason as to why we might be considering use of shedding analysis in when we're looking at such new products. Why it hasn't been done before is potentially because, as I mentioned, the latest scientific discovery is not yet reflected in these guidance documents. And this is also potentially reflected by one of the comments that was mentioned in the document that I found interesting, which said that peptides, which are basically fragments of proteins, are not expected to be a form of infectious agent or transmissible between individuals. And it's mentioned as they're never participate in that, and that not necessarily might be true. We are now learning, precisely thanks to exosomes, because we are learning that exosomes, and we'll be discussing that in the future, could cause immunological reaction, and they can transfer proteins and peptides as well. So you can see that might be the reason why exosomes and breath are not yet considered as a possible option of shedding material, and we might need to wait for the updating of this type of information in order to have such studies performed in the future. All right, but then now, and also if shedding does happen, or sorry, if shedding studies do happen, finally the document also mentioned what kind of information should be analyzed. I thought that was also interesting. So of course, what should be analyzed is how many patients were shedding the material. That's number one. Number two is how many samples were contained the shedding material, and which samples never did. How long does the shedding occur and take place, and when is the peak of shedding? So you can see a lot of different information. And then finally also the quantity of shedding. So shedding product that could be causing something like the immunological response. So you can see a lot of information could be potentially gained from proper shedding experiments that should be done when you're dealing with potential novel products that could be transmissible. All right, so FDA does not recognize shedding of exosomes via breath. Now, is there any scientific evidence that could happen? And the answer is yes, but it's very, very new. It's basically nascent information. It's just coming to a forefront, and this is probably why we are not yet ready to consider this as a standard, and why perhaps in this mass vaccination program we might have inadvertently discovered this because of the constant observation that women who were exposed to vaccinated partners had their menstrual cycles affected. So this just simply might have been discovered simply by accident, because so many individuals around the world were vaccinated so rapidly that it allowed very rapid clinical symptoms being apparent, and why the populace people basically started suspecting what is going on. Could there be shedding, and what could that include? So now let's talk about shedding of exosomes via breath. The very first paper that I was able to find that discussed this in any way was only in 2013. And in that paper, what the authors wanted to do is they wanted to look at, let's show you that. What they wanted to look at is Can microRNAs be used as biomarkers to differentiate healthy individuals from individuals who have asthma or tuberculosis? So what are microRNAs? Hold on. What are microRNAs? These are tiny RNA fragments. They are not used to produce proteins at all. Rather, they are released by cells, or they're also used inside the cells. They are released via exosomes. It's been very well known by that. And this is one of the ways how the body can regulate the mRNA's usage inside the body. So basically what we're talking about is when you produce different mRNAs that are supposed to be used by your body for the purpose of producing proteins, microRNAs can start influencing how much, if at all, the mRNA templates will be used. So it's just another form of regulating the entire system. So they're very powerful, these microRNAs. And the question is, can you use microRNAs as a biomarker, basically a way to differentiate between one individual who's sick versus individual who's healthy? And these authors wanted to look at microRNAs, and they did indeed show that there were different microRNAs in people with asthma and tuberculosis. They found 11 different microRNAs that were expressed differentially. That means when we talk about gene expression or use of RNA, production of RNA, expression refers to the quantity that RNA is being produced at. So when we talk about differential expression, we're talking about how much different is one type of individual producing RNAs versus another type of individual. So that's differential expression, and they were able to show that people with tuberculosis and asthma were showing different RNAs, and some of them were previously already linked to both indeed asthma disease as well as inflammation. So this suggests that perhaps inflammation might be problem in these in a collective problem amongst these respiratory conditions. But here's the key one. What the authors also showed is that the majority of the microRNAs that were being isolated from these individuals via breath, and this is the key, they were looking at breath. I forgot to mention because specifically we're talking about breath. So they basically look at the breath for the first time ever. Because why breath? Is because obviously breath is easy, technically non-invasive and easy to capture, and also, of course, it brings the biological material that comes from, well, your lungs and your nasal cavity, right? So it has the biological material directly from the tissues you're interested in studying. And the vast majority of the microRNAs that were being captured were inside those exosomes. So they're able to confirm they were exosomes because exosomes can also have proteins on their surface, just like coronavirus has a spike protein on its surface that sticks out, hence the name, right? Coronavirus. Exosomes can have the same, and they can have proteins on their surface and they can stick out, and they capture these exosomes via one of those proteins that is very commonly observed amongst exosomes, and they used antibody that was specifically made to recognize that particular protein and capture that protein, and they were able to show, that's how they were able to isolate exosomes from breath and show that there are different microRNAs. Now, that was the first mention ever that exosomes are being isolated from breath. Since then, the only other paper that I was able to find was in 2020, so only just over two years ago. And it was the first time ever that they were able to characterize exosomes from breath. So the first paper I just mentioned a moment ago was the first time ever they were able to isolate microRNAs via exosomes from breath. This paper I'm just about to talk about briefly was the first time the authors characterize exosomes from breath. So how do you, by the way, capture that? You breathe into this this column that freezes the breath, and this allows you to condense everything, and then they use filters that specifically worked to filter and isolate exosomes. As I mentioned, exosomes are specific size. They're similar to coronavirus size, and they were able to actually filter them from all of the other larger size material that came from breath. And these authors basically showed that indeed exosomes can be isolated from breath. And they used microscopy. They used electron microscopy, two types of microscopy that allow you to see minute material, and from the images that they captured, these are pretty much like photographs that you can take of super small, tiny biological material, and indeed you can see exosomes being captured from breath, and they were able to show that these exosomes are the same size that you expect exosomes to be found in other tissues. We're talking about somewhere between 50 to 150 micrometers in size. As I mentioned, that's very similar to coronavirus's size. Okay, very, very tiny, but that's basically how cells can release them and how cells communicate. These same authors then showed that they can use different techniques to capture these exosomes by the same protein marker on the surface of exosomes as the guys in a previous paper, and they were able to basically confirm, hey, we are indeed capturing exosomes from breath. This is legitimate. Now, I haven't found any other papers exploring this since 2020. So this shows you how new this information is, and why scientists in general might be at the moment very reluctant to state that it could be possible to shed vaccine material via breath, because this is just simply something so new that overall scientifically we don't have a very good concept of. But we're going to be talking about that in the upcoming two videos. What kind of exosomes are indeed isolated from the vaccinated individuals, and what kind of exosomes are being isolated from COVID-19 patients. And clearly the big question is, have exosomes of any kind been isolated from vaccinated individuals? And I have not been able to find anything. I would suspect no such studies have not been done yet, because these are obviously very rarely done scientific studies. So I doubt we have anything, but maybe I missed something. And it would be fantastic study to do, because this would help us understand, is shedding of vaccine-related material possible via breath? And this would help us. But we'll see what can be shed from exosomes of vaccinated individuals. So please stay tuned. Well, I'm gonna end here. This is at least the first introduction. Technically, theoretically, shedding is possible via breath, but we need a lot more evidence. I say that a lot in many of my videos, because we're always talking about the latest science, right? So I want to say thank you for always the support. Thanks for all the questions. You can see how this type of questions that the audience is asking eventually leads to scientific studies, and what I specifically analyze. And when it came to exosomes, as I mentioned at the start of the video, it's been a long time study, but I interspersed with study of clotting in COVID-19 patients, because that's another one of the more common questions that I've been receiving. Hey, is there a possibility of strange clots indeed being identified in individuals, like we have been seeing in some of the unconfirmed videos online? So stay tuned for that information as well. So thank you for always asking questions, and that's keep it coming. And I look forward to seeing some of you at my upcoming COVID Q&A events, where you can ask any questions as well. Those are live, and that's also where I obtain a lot of scientific material. So thank you for that. And finally, thank you for the kind soul who has made this toque for me for my travels. So that's really cool. I really appreciate that as well. It has been keeping me warm on this trip. Bye, everyone, and I'll see you next time.